Yeah, so we're going to go into NMR in some more detail, and we're going to have a breakout sessions with groups right at the start of class, I guess, pretty soon. So I just want to make sure you guys are aware of that if you're working on other things. But today we'd like to focus more on the instrument components and some of the biophysical applications and not just the theory behind it. But I do want to recap, since it has been an entire week since we first talked about NMR, that we introduced NMR as a absorption process in the radio frequency range by nuclei in a magnetic field. Where this radio frequency range that's used in NMR is less than 10 to the 9th hertz, or that would be wavelengths of greater than 0.3 meters or so. That region of the electromagnetic spectrum with the addition of this magnetic field. So we went over both the quantum descriptions of this, where we have an energy diagram here showing that as you increase the strength of the magnetic field, the energy difference between your two spin states increases. So that improves your sensitivity of your NMR measurements, and is why people build giant magnets to do NMR for very sensitive work. We also talked about the classical description, where you can view, okay, if you have quantum mechanical descriptions of the spins of nuclei in two possible directions with or against the magnetic field, that from the classical description, you can then sum the distribution of these states and get a bulk magnetization of your sample that will change and decay over time. So we talked about the decay processes, T1 and T2, of those nuclei spin after you turn off the magnetic field. And that the reason NMR is such a powerful technique is that it's very sensitive to environmental effects of neighboring nuclei or electrons. So this is giving you atomic level resolutions. And then we also briefly mentioned that there's electron paramagnetic resonance, which is a similar phenomenon using electron spins where it has to be an unpaired electron. So NMR is more common because you have nuclei, you have protons, you have carbon, you have nitrogen in molecules naturally, but it's very rare to have an unpaired electron, so you have to label your samples in contrast to NMR. And also that EPR is in the microwave region of the electromagnetic spectrum. So just a quick recap of that's the topics we discussed last time. So today, getting into instrumentation and applications, I have a block diagram here of an NMR, and we're going to go over this in more detail, but I want to mention that at least this block diagram here is showing Fourier transform NMR. And historically, NMR was performed at a continuous wave method before the 1970s, but then with computation, Fourier transform NMR improved the sensitivity greatly, because as you pulse your radio frequency, you can then have higher resolutions. And so we're going to go over this in more detail, but you have with your radio frequency for Fourier transform, you're going to pulse it at megahertz frequencies. Let's see here. And then as you detect that, if this is a, I guess, excitation, the detection is going to have this sinusoidal decaying wave that's called a free induction decay. So you hear FID, that's what is this discussing, seeing this decay over time due to the fact that you're pulsing your excitation. But today for the breakout groups, I mentioned it's going to be at the start. I'd like to, instead of explaining all the components of the NMR, I'd like you guys to look into a little bit of detail and discuss these components. So within each breakout group, I'd like you to decide, okay, with a different source. On Canvas, in the resources section, I have these specific pages noted for the leak book in chapter 5, or in the Scoop Collar Crouch book, these pages talk about the instrument components for the NMR. And then also you can do general searches on Wikipedia or looking at the vendors. So within your group, each of you guys take a source and look at a corresponding component within your group for about 5-10 minutes, meet back up and discuss these, and then assign someone to report back what you guys found or agreed upon. So group 1 will be focusing on the sample and sample conditions in the holder here. Group 2 will just investigate what type of magnets are used and what conditions you have to use for the magnets. Group 3 will look at the radio frequency source, and you can see that the coil here is important for both the radio frequency source and the detection and where the sample is. That's why all these colors are overlapping. And group 4 will look at the radio frequency detection or anything specific about that. So do you guys have any questions before I break you guys out into the groups? Okay, so let me just find the breakout groups. Okay, and give me 1 second. I'm also, I think Dan, Colin, and Rose, you guys have all taken like NMR classes, right? Is there anyone else who has? Okay, because I'm going to put you guys in separate groups. So, okay, I'm going to open all the rooms now. Any questions? And I'll try to share the screen back in this main room. So I'm going to open these guys, and a little bit after noon, I'll send you guys a message to meet back up.
Oh.
You always tell which groups need more time. Once the breakout announcement is shared. Okay. You guys can see the screen, okay? Okay. So group 1 with the sample, what do you guys find out?
Okay, I guess I was chosen to talk about this. So I have a fair amount of experience in actually doing this. So this is mostly firsthand accounts of me preparing samples before, but basically with your sample preparation, a lot of these NMR samples are done in, you prepare them in small little glass tubes, kind of similar to what you can see in the schematic on the screen there. And you'll typically analyze either solid, liquid, or gas samples, but gas specifically is a little bit harder to do. It requires special sample tubes that you need to use in order to flow your gas in and out of the tube.
Yeah, essentially a small number of molecules, like lower number of molecules, that makes it hard to.
But for solid and liquid samples, which is what I have most of my personal experience in, you're typically gonna put those inside of your NMR tube and then dissolve whatever solid you have or liquid in some deuterated solvent. And the deuteration of the solvent is important because otherwise you'll end up picking up those protons in, like, for example, a water molecule versus a deuterium oxide. And then you typically will have a certain amount of sample that you want to put in there. I personally tend to put around 10 to 15 milligrams in, but we found a reference that said about anywhere from 5 to 25 milligrams is good for this. In my personal experience, sometimes it is better to have more than less sample because you may have problems with actually trying to lock onto the magnetic field and shim the magnet, which makes it harder to actually obtain a spectra. And similarly, there's a certain amount of solvent that you want to put in there. There's a cute rule that some of us use called the 3-finger rule, where on a lot of these NMR tubes, because they typically have about the same diameter, you can use just 3 fingers stacked up, and that's about the height of sample that you want. That's important because if you underfill or overfill, then your sample width will actually be not centered in the magnetic probe, and if it's not centered, then your spectra, the peaks will end up getting, they'll get shifted a little bit, or they'll get kind of side humps almost. And it could also, a less solvent can make it hard to additionally lock on to the magnetic field and shim the magnet. After that, you typically put, you'll cap off your sample and put it in this sort of spinner, which kind of has a hole in the middle. Pretty much all, and anytime you're running an NMR, you'll see these spinner these spinners, and they typically come with like some tube or something that will guide you as far as where on the actual sample tube that spinner should be located, as far as like high up. But it's kind of like a donut almost that goes around the sample tube, and that spinner has usually like white and black markings kind of around the edges that will indicate, I guess, if you're spinning your sample in the magnet, it will tell the magnet, how fast it's going, or things like that. And yeah, then you typically insert it into the top of the instrument, and there's usually an air compressor or something that will shoot air to hold your sample, and then it'll kind of lower it into the probe. And I guess that's all I can think of about a sample, unless someone else from the group wants to jump in.
And do you guys know why the samples are spun?
So I mean, they're not always spun necessarily. Spinning them, I mean, it can both have an effect on, I guess, a small effect on the homogeneity of the actual sample, but also helps that out the I guess the homogeneity of the magnetic field as well.
That's a very nice detailed description, and it's useful because you use them. So
Yeah, I've done a fair amount of this.
I don't do much NMR, so I appreciate people who have can provide those details. Okay, so let's move on to group 2, describing conditions with the magnets and components there.
Okay, so the magnets, at least when we mostly looked at for a modern NMR system, it's typically a solenoid coil. So just a a coil of metal wire, and you run the electric current through, and that's how you get your magnetic field. I guess in terms of the wire itself, it's typically some some conducting wire, with niobium, titanium, or tin, or usually an alloy of all those elements. And then of course the feature of the NMR is that you for superconducting in NMR, you have to reach very low temperatures, around 4 Kelvin. So to reach that, they surround the magnets in liquid helium, and then they actually surround the liquid helium in liquid nitrogen to protect it from the ambient environment of the room. And superconducting magnets seem to be pretty common these days for NMR.
Cool. And did you guys read anything about shimming?
Oh yeah, actually, yeah. So shimming I guess it's like a technique to reduce noise or improve the signal by running another smaller magnetic field, or running a current, I guess to help with interference.
Yeah, fine tune that magnetic field. Yeah. So that's an additional complexity. Yeah. That's pretty good coverage. And you'll see with liquid helium, I know someone who runs an NMR core, and they're always like, I don't know if you guys have heard about how like there's helium shortages and we have to mine helium, and it's a limited resource. So when you're using those helium balloons, you're taking away helium for our NMR measurements. I've heard people say for cooling those down. Cool. Any other comments about magnets or questions? Okay, let's move on to group 3 then.
Yeah, so we did a little bit of a less detailed approach, but basically from what I understood, there's like some radio frequency generator that generates like a known frequency, and then we have a frequency synthesizer that can produce a range of frequencies based on that one, I guess, reference frequency. Then we have a gate that basically controls the pulse widths. And then from there, our signal goes into this power amplifier that will amplify the power, unsurprisingly, to maybe 50 or is it 50 to 100 watts? I mean, the width of the pulses kind of corresponds to the range of frequencies if we like Fourier transform that square wave. So I guess if we have narrower waves, that corresponds to a wider range of frequencies. Yeah, I don't know if anyone else in the group wants to add stuff. Okay.
Yeah, and to actually impart that signal to the sample, like there's a coil of wire around the sample. So that's used both for the source and the excitation, like what's shown up here, and then also for the detection if group 4 wants to present what they found.
Okay, can you hear me?
Yep.
Okay, so our part is the radio frequency detection, and we can see there's 3 components. There's the coil that was used before for sending the RNF. It's also to transmit, is also used to receive. There's the RNF amplifier, and there's the phase sensitive detector. So what we do is usually we know that we put our sample in the magnetic field to kind of like have all spins going in the same direction. Then we apply this pulse that could be at 90 degree or 180 degree, just to kind of like shake that sample. And this is done by the coil, the transmitter coil. The idea now to detect the signal is then we turn off that pulse, and then as the spin is going back to its original position under B0, it emits like another RNF. There's another energy coming out at kind of different frequency than the transmitted. For sure, it's lower. As it processes to go back to its original position, it loses energy. And that energy is kind of like like motor force that is also switched into it's picked by the coils. So this is the idea of the coil, is to pick that RNF frequencies. So once these RNF frequencies are picked up by the coil, it's sent to the preamp. The main job for the preamp is to kind of like clean the signal, the detected signal, from the noise. So what they do is they ensure that the final signal to noise ratio is dominated by the coil and the sample and not by the electronic noises. So, and then the preamp send the signal to the phase sensitive detector. So the idea in the phase sensitive detector is that even though these RNF frequencies are detected, to kind of be able to digitize them, you need to compare them to like a reference frequency. That reference frequency, I'm not sure if it's a Larmor frequency or it represents the TMS that was explained last time. But it's like a reference frequency that is used, and we compare our signal to that frequency, and what we register is the difference between these two signals. And this difference between the frequencies is digitized and stored. And there's like multiple there's I would say equations for that, which we're not going to go deep into that, but and then these signals, after being demodulated, the signal goes down to audio frequencies, and the signals are then sent to like analog or digital converters and being acquired by the computer and processed and stored. This is what I have for this part.
Yeah, that's a very nice description. And I drew these arrows when you were saying about the phase sensitive detector of like what the reference frequency is. So that would be the frequency that you're inputting, your input frequency as well. So this phase sensitive detector will, similar to like a lock-in amplifier, you're going to want to compare, have it from what you're sending in and then what you're detecting out.
Okay.
That's why I put for LIA, that's lock-in amplifier. But that's a nice description there and concise.
Okay.
Cool.
Thank you.
Any questions about all these different components? Those were really nice descriptions from you guys. Okay, cool. So we'll go over some specialization and applications now of NMR. Rose was mentioning 90 degrees and 180 degrees with the pulses that are sent in, and these are specializations of how you pulse your excitation with Fourier transform NMR. So if you hear, this is sometimes called like echo pulse sequences, and that just means you're going to be using multiple pulses timed differently to help enhance or enhance your signal, improve your signal to noise ratios. So this can be used for signal averaging, and also dephasing. So with these diagrams here, this would be like the most simple pulse, where you just have one radio frequency pulse going in and looking at that free induction decay here. But if you want to get more specialized to get higher sensitivity and better signals, you can see in this diagram here, you can have the timing separated by different phases. And that's why the phase relation here is why this isn't listed in degrees. So your signal here, this TE, corresponds to the echo time. And you can relate this, your signal can then be related to some proportional constant. Based on this rho is going to be a given density of nuclei. And the Fourier transform of this would then, of this type of pulse sequence here, this echo pulse, would be 1 minus the exponent of your repetition time. So your repetition time is how often you're sending these first pulses. And divided by these relaxation processes that we discussed, your T1 from last lecture times the exponent of your echo time over T2. So this helps improve your ability to look at that relaxation process, the T2 relaxation. So realize that using different echo pulse sequences can improve and change your sensitivity there in a more specialized form of running your NMR. So that's just another mention of, I guess this relates to, on the previous slide, like the radio frequency excitation, that it can be specialized with different forms to relate to that Fourier transform. So that's a little bit more detail about that setup. But I want to move over to talking a little bit more about biophysics, since this is a biophysics course. And for those of you that use NMR, or also in our discussion how you can use this like in solids or for liquid samples as well, what ideas do you guys have of why NMR would be used instead of, for structure, instead of the methods we were talking about last week, like cryo-electron microscopy or X-ray crystallography, or even some of the fluorescent methods? Any ideas or thoughts? So like thinking back to the recap, or the fact that this is sensitive to the electron density and nuclei around certain protons or certain atoms, like what type of resolution can you get with NMR? Spatially.
So are we talking like nanometer, or like on the order of the SEM sort of, on sort of,
So yeah, you can get nanometers, you can even get sub-nanometer information. So this is an atomic level resolution technique. So that's why this is like better than the fluorescence techniques, where it's harder to get down to these atomic level scale resolutions. But like crystallography and cryo-electron microscopy, they get down to that atomic resolution. So why would you pick to use NMR over crystallography or cryo-electron microscopy? Yep. So Daniel's saying that the sample prep. So you get these atomic level resolutions, but you don't require a crystal or a static sample. And because you can do this in a liquid phase, a very important thing that you can get from NMR that you can't get from crystallography methods is dynamics. So you can see changes if you pump or probe your sample in specific ways, if you introduce new things to your solution, if you can see these type of dynamics and fluctuations at different time scales, and we'll discuss that. So that's the key things of NMR, atomic level, but dynamics as well. So that's why it's a very powerful biophysical technique. And getting into some of the specific samples that NMR is used for, if we get this working, we'll start off with talking about lipids. I have these figures are separate, so I'll go over those in more detail in a little bit. So NMR for lipids is used for looking at more of like the surroundings and the order. It's not used anymore for chemical identification. And by chemical identification, I mean like what lipids are present in my membrane or in my vesicle or in my bilayer. So if you want to understand what lipids are present, mass spectrometry has replaced NMR for chemical identification of lipids. And we'll be discussing mass spectrometry, I guess, later this month. And we'll go into detail there. But instead, it's more focused on like, how is it organized? So for example, this data here is showing the peak location and the width for, I think these are vesicles of different sizes. So you can imagine, I mean, this is a model study of vesicles of one type of lipid, but you can think even if you have a more complex sample where you're trying to understand maybe self-assembly of different mixtures of lipids, or if you're able to extract organelles somehow or cells, that you can get at, okay, the width of this relates to the size, and then the heterogeneity would relate to the different peaks that are present. The NMR, this is a Lamar frequency shown here that also can relate to what's the structure of a planar lipid bilayer. Has signals that look like, or the frequencies here that for looking at phosphorus and the protons, compared to if you have a hexagonal structure, these signals the frequencies clearly change here. So the organization structure is key here, and it can also tell you a little bit about like the environment of like how hydrated are your lipids, what's the solvent or how much water is around it. And also, given these structures, NMR is used in solid state forms, and also magic angle spinning, which we could, which is when you're doing a solid state measurement of NMR. If you measure it at a certain angle, you can reduce the amount of noise or background that's present there and get higher signals. So mostly organization with looking at lipids. Getting into proteins, I have some figures over here of different scales, different proteins, and NMR to understand the 3D structure of proteins, getting into some of the history. And like if you go in the protein data bank, many of the deposits are determined by NMR. And the first time of measuring a protein by NMR wasn't long after the discovery of NMR. So if you remember last time we talked about that NMR was in the 1940s, that in 1957, 1D proton NMR was used to look at ribonuclease. But traditionally, 1D NMR is very challenging to get structures of proteins, and I'll show this on the next slide, but here's an example of a 1D NMR of just the histidines in this very small protein. So you can see this, these have broader peaks than, and this is just looking at one amino acid within that protein. So if you, I guess I'll just show on the next slide here, that if you take and look at the protons in an entire protein, it's quite messy and it's hard to identify like what's going on here. So while this was measured in 1957, it didn't really become a routine technique to look at proteins that much until later. when there were instrumental improvements. So better magnetic fields, cooling the samples, getting very specific with the isotope labeling. Similar to this guy up here, if you can just look at protons or nitrogens or carbons in specific sites, that will help you identify what you're looking at. And the main key advancement was multidimensional NMR, which we're going to discuss in more detail in the next slide. But this plot here in B is showing if you're able to look at two different nuclei and look at their cross peaks in two-dimensional NMR, that can help you greatly identify, okay, what specific atom am I looking at in the protein as opposed to those very noisy broad peaks in a one-dimensional NMR. And the development of using multidimensional NMR to study proteins was actually, since it is Nobel Prize week, the 2002 Nobel Prize in chemistry was 50% of it was awarded to Kurt Butrick for being able to look at the 3D structure of proteins using multidimensional NMR. The other 50% went to mass spectrometry, which I mentioned, we'll discuss later in the semester. And I also want to mention that when we're talking about the benefits, the type of dynamics in proteins and also other biomolecules that NMR can access can range from pico to nanosecond dynamics of looking at spin relaxation phenomenon. You can also get to millisecond to seconds when you're looking at how line shapes change over time. Or relaxation dispersion. And you can even get to minutes to hours if you introduce hydrogen and deuterium exchange. You can exchange those protons out if you flow a solution in to see reactions that exchange or ligand binding. That it's very impressive of the time scale, the order of magnitudes of time scales that you can probe with NMR looking at these proteins. So an example with looking at like ligand binding, you can get high resolution spatial information. So that's what this figure is here. So an apoform versus a holoform of the protein means without or with a ligand present. And you can see the ligand in this protein is this purple guy over here. And then these red and yellow and orange balls are showing the degree of change in distances for different residues, where red is indicating like a large degree of change in the position versus yellow where it's more static. So this is giving you, again, that atomic level resolution and structural changes. With this experiment, they probably did measurements of two separate solutions of the apo and holoforms of the protein. But again, this time scale process, you can look at these changes over time as you introduce a ligand to your solution. And similarly, you can look at proteins associating with one another. So there's lots of dynamics going on there. So getting a little bit into more detail of this multidimensional NMR. If you read the NMR literature, you'll hear these referred to as COSY techniques, or always with this SY, where COSY stands for correlation spectroscopy. And you reduce overlapping peaks, or I guess the overlapping peaks in 1D NMR make this a challenge. So if you look at, if you increase the dimensions, you can look for hotspots that are molecular signatures. of the structure. And this has to do with the transfer of spin between different nuclei. So that's what this quantum mechanical description here of where this is the focus of the 2D NMR of that spin being transferred from one nuclei to another. So I have an example here of looking at, this is homonuclear multiple dimension NMR, where you can see we have proton NMR on both the X and the Y axis here. And this can correspond to that spin transfer of nucleotides, residues from one another that you can see here, that they're able to actually label these with specific protons on either side of this RNA aptamer present. So this is what the type of data looks like here. Some drawbacks of doing multiple dimensional NMR is that it takes more time to collect. It's more like hours, so it's not as routine. And this transfer of spin that I mentioned is, you should be familiar with this term of nuclear overhouse effect. Or is called noisy. And the NMR people can, more familiar with this, can correct me if I'm pronouncing these wrong.
Yeah, it's nosy.
Sorry?
It's nosy.
Nosy, okay. And there's lots of these SY techniques that you can see here with the homonuclear, where you're doing both protons, that works well for nucleic acids since they're relatively small, but with proteins, it's still noisy or difficult to interpret. So for proteins, you have to do heteronuclear nosy, which I guess is sometimes called HOSY. And there's also rosy, which is rotational frame. There's lots of variations. And again, just like many biophysical techniques, there's an alphabet soup of, as you make slight changes, you call it a new technique there. So I think this is best illustrated on this slide. Yeah, let's see in the chat. Yeah, total correlation, diffusion ordered. Yeah, so you can look at the diffusion, all of these. So I think this figure better illustrates of like why you want to do the heteronuclear multidimensional NMR for a protein. So this is ubiquitin. It's a relatively short protein that's in all eukaryotes, and that's why it's called ubiquitin, because it's ubiquitous in eukaryotes. And you can see again, it's very difficult to interpret. If you have small changes in the structure of the protein, are you going to be able to resolve that with these broadened out heterogeneous peaks? And even if you do the homonuclear, this is H1 versus H1, proton versus proton, for still a very small protein, this is very difficult to interpret. There's a lot of overlapping signal, but this cleans up so much better. This is the same protein of ubiquitin, that you can find these individual peaks. If you have a small structural change, it'll be very clear if these peaks shift in a certain direction or disappear or so. And with those pulsed sequences and the echo arrangements, this is also being shown with the time sequences shown down here of how you're measuring with different pulses for a transforms of looking at the proton and nitrogen signals with certain delays between them. So again, this is why there's entire classes devoted to NMR, because we can't really cover this in one class of all these pulse sequences, but I just want to mention those to be, so you're familiar with them. In the interest of time, I'm just going to mention that MRI is an imaging form of NMR, and that we're going to discuss this probably like one of the last classes of the semester when we are going to be scaling up and not thinking about biomolecules, but focusing more on organisms and larger scale structures. So we'll get into that later, but I just want to mention that since we are talking about NMR, that these are taking advantage of the same physical phenomenon of probing nuclear spins with radio frequencies. And then finally, I just want to, since I do want to mention a little bit about how EPR, what that data is, how the instruments work. And you can see that this is what a typical spectra would look like for EPR, or specifically here, this is a commonly used radical label that's measured in EPR. And you can see that the signal looks quite different than what we have been showing for NMR signals. And this is called the hyperfine splitting, which is showing how the electron spin interacts with neighboring nuclei. So again, this is going to give you atomic level information because it's looking at the local environment of this very specific one electron that's present. If you want to use EPR, the instruments are categorized on these different bands that correspond to the different microwave frequencies that you're studying, and the stronger your magnetic and the magnetic field that you're using, the stronger the instrument, the higher resolution that you're able to get. And by resolution, I'm meaning like the spacing of the peaks here. So I'd say traditionally you'll see Q band quite frequently. With the instrumentation, W band and higher is going to be using like the superconducting magnets, while lower than that, you can use electromagnets. This is just a picture of what an EPR looks like, shown down here, where the magnet is and the microwave cavity in the sample is shown here. And that's really like this type of setup where you see this structure, I think is, if you're walking through a room and you're not sure what an instrument is, if you see the sample area that looks like this hooked up to some liquid nitrogen, then you can tell that's an EPR instrument. So we do have one in the biophysics department over in the medical school. I think it's associated with one PI's lab in the biophysics department, but it's part of their core facilities. And then in the state of Ohio, actually the University of Miami has an entire facility dedicated just to EPR, and they have 6 different EPRs there with different bands. They even have a benchtop EPR that's available. So just letting you know that those are around. We don't have a ton of time to go over all of the instrumentation, but things I do want to point out is that the microwave generator is usually, historically it was done with a vacuum tube with high gain, but now how it's being used is a gun diode. And this is an N-doped semiconductor that can produce microwave frequencies. But the unique thing, you can see that this is traditionally done in Fourier transform as well. So that's why you see like this frequency counter. There's going to be up here, this is like a reference line. So similar to having like a two excitation bands in UV vis, you do the same thing with this microwave resonator. The unique thing here is that you have a circulator that sends signal down to your sample within the magnetic field. In this sample, it's actually a cavity to help enhance the number of interactions between your microwave and your sample to help increase your signal to background ratios. And after it resonates in that cavity, it's sent back, and the circulator is able to send just the microwaves that interacted with your sample to then be locked in and detected compared to your reference line here. So those are some unique aspects of EPR instrumentation compared to NMR, that you have a resonator cavity and this circulator that's present. So I just want to mention a little bit about the instrumentation for EPR there. So again, you get same references and resources. And that's most of what I had. Any questions at all?